All right, welcome everyone to our Quercus Quest Campaign Kickoff webinar. We are very excited to have you here today to learn all about this new campaign that we're kicking off. We are going to be having you all on mute throughout the webinar today. There will be a chance at the end for questions, so we can either have you unmute and ask your question, or you can just put those questions in the chat and we'll make sure to get to them at the end. We're also recording this webinar right now, so we will be sending out the recording in a follow-up email probably later today or tomorrow. So the plan for today is to go through this list of things here. I should say I am Erin Postumus. I'm the outreach coordinator here at the USA National Phenology Network. I'm also joined by Samantha Brewer, our volunteer engagement coordinator, who's going to be helping in the chat and helping to answer the questions. And we also are joined by three of the researchers that are behind this campaign, and they're going to be explaining all about the bigger project that this campaign is part of. So we have Andrew Hipp, Ian Pierce, and Janine Cavender-Barris here with us. So they're going to be sharing an overview of all of the research, and then we're going to be getting into an overview of the USA National Phenology Network and Nature's Notebook. We'll go through how you can get started with this campaign and explain some of the training materials and then the resources that are available to you. And then we'll get into the Q&A at the end. We invite you to put where you're calling in from and your name in the chat, just so we can see all the places that you guys are calling in from. And with that, I will turn it over to Andrew Hipp, who's going to talk about the research that's behind this campaign.
Thank you very much, Erin. I'm very glad to meet everybody. My name is Andrew Hipp. I'm the plant systematist and herbarium director at the Morton Arboretum, and I'm just one of a large team of researchers working on this project. Here with me today are Ian Pierce and Janine Cavender-Barris, as well, each of whom is going to be presenting on a different component of our research. I'll kick this off, see if I can successfully share my screen. Am I successfully sharing my screen?
Yes.
Yes, I am. Thank you. Okay, let me minimize that, and I'm going to do a slideshow. Are you still seeing my slideshow? I cannot see you, so if someone could say yes or no, that'd be awesome.
Yes.
Thank you so much. So this project is about, this is an international project with a team of US collaborators and a team of Chinese collaborators asking a very basic question, which is, how does hybridization, gene flow among tree species influence the communities of insects and fungi that live on those trees, as well as influence how well those trees adapt to regional variation in climate and soils, all the things that trees need to live. That group of interbreeding trees is called a syngamion. We'll see this word show up a couple of times. You don't have to know this word to understand any of the things that I'm going to be talking about today, but it's a handy word to know. So this grant investigates how oak evolutionary history, the phylogeny of oaks, that branching set of relationships that ties all the oaks back to their most recent common ancestor about 56 million years ago. How that evolutionary history, then genetic diversity within oak species, and hybridization between oak species influence the traits of those oak species, as well as the communities of organisms that live on oaks. So we're asking about all these interconnected bits of what makes an oak, both its evolutionary history and how it fits to the environment. And then how that in turn shapes the organisms that live on them. Things like the leaf endophytic fungi. This is fungi that live inside the leaves. Mycorrhizal fungi. This is fungi that live on the oaks. Roots, on the roots and help oaks get nutrients. And gall wasps and leaf herbivores, insects that live on the leaves. So the Eastern North American white oak group is what we call a syngamion. This is good species across their range in spite of a long history of gene flow. So when we look around the edge here, we have white oak, we have swamp white oak, we have bur oak, we have post oak. All of these oaks exchange genes to a certain extent. These lines between the leaves indicate what we know about gene flow between these two species based on our molecular data, if the lines are very thick, or based on literature reports if the lines are much thinner. This movement of gene copies between species when species pollinate one another is likely a key to the ecological and evolutionary success of oaks. Now, we had a question before this even started. Someone emailed in a question about why we're only focused on white oaks and their relatives. The reason is that white oaks and their relatives can all interbreed. They are one syngamion. Red oaks and their relatives can all interbreed. They are another syngamion. We could have focused on both, but we chose to just focus on one in depth, which is the white oak group. Partly because there's plenty of them, they're doing a lot of hybridizing. Partly because one of the most widespread, in terms of range, oaks in North America, Quercus macrocarpa, the bur oak, is in the white oak group. Bur oak stretches from Manitoba all the way down to the Gulf of Mexico, and from the Black Hills all the way out to Massachusetts. So it's an incredibly widespread oak. So for those reasons, the white oaks turned out to be pretty good to focus on. Also, they've been studied a lot in the past. So by investigating flowering and leaf out phenology of bur oak and co-occurring white oak species with which it can hybridize, you will be helping us to understand species potential to cross-pollinate and the timing of resources for the insects and fungi that depend on oaks. And these are just pictures of the range maps of bur oak, Quercus macrocarpa, white oak, and the other oaks that we're focused on. In these pictures, the shaded areas represent what's known of their natural range, and the triangles are all individuals that we've sampled in the past for molecular studies. Your work has the potential to give us information now about phenology for these species across much of their range. In addition, the data you collect will be compared with phenology data that we'll be collecting in 3 reciprocal transplant bur oak experimental plantings. What do I mean by that? These reciprocal transplant experiments are experiments in 3 places. This is a project that was really spearheaded by Janine Cavender-Barris, who's going to be speaking next. One of the gardens is at her home institution in Minnesota. One is at Heather McCarthy's home institution in Oklahoma. She's another collaborator on this grant. And one is at my home institution, the Morton Arboretum, here in Lisle, Illinois. Trees from all 3 regions were collected as acorns from mother trees and planted out to all 3 regions. So this allows us to see how oak phenology and other traits, including the fungi and insects that can live on oaks, change as oaks shift in range. By combining that range-wide data with these garden data, we'll have a really good understanding by combining the work that you're going to be doing with the measurements that we'll be making in our gardens and that we'll also be making in the field as well as we do our field work, in understanding what the contributions are of genetics, of evolutionary history, of hybridization to all these opportunities for oaks to both exchange genes and to host organisms that depend on oaks. Okay, now in addition to these white oaks, we have added in 2 other white oak species that have been really heavily studied by Janine Cavender-Barris and her crew. So I'm going to stop sharing and hand off to Janine to talk to you about just one of these species pairs that's really exciting. So Janine, alley-oop.
Thank you so much, Andrew. And I'm going to talk about the live oaks. Can you see my screen?
Yes.
This is the famous angel oak in South Carolina that itself makes up a whole ecosystem, and it's a coveted tree in the South. It is a white oak in the broad sense. It can interbreed with the white oaks that Andrew has been talking about, but it also is its own lineage. And there are only 7 species in this lineage. And they are beautiful trees. In the southeastern US, they go down through Mexico and into Central America. There are two species in the southeastern United States. These are southern live oaks, and the one that's most famous that you see in the movies is the southern live oak I just showed you, Quercus virginiana. And you see it has these glabrous leaves with smooth margins. And then its sister species, its closest relative, is the sand live oak, Quercus geminata. And these look very similar, but their leaves are a little different. The sand live oak has this rugose venation, quilted looking leaves. And as if you look on this graph, you see that it occurs in drier habitat. So the x-axis is soil moisture, and this species occurs in drier, slightly drier environments, whereas Quercus virginiana lives in a broader range of environments, and it can get fairly wet where Quercus virginiana lives, but they often occur together. So there is this range where they occur together too. And they do share genes, and they do interbreed. And so one of the longstanding questions among these two species, there's a third one too that's actually very short, that it's the Quercus minima, and it's a fire adapted species. But let's just talk about these two species. A longstanding question has been, does the length of the season influence their flowering time in a way that farther north, they can't separate their flowering time, and they're sort of forced to interbreed more. So they hybridize more in the north. Whereas farther south, they can spread out their flowering times more, and they can separate flowering a little more, so they stay better, more independent, and don't share genes as much. It's a question, and it's a hypothesis that the shorter growing season farther north means that they probably should hybridize more. And maybe we see some evidence for that, but we don't actually have the data. So here's a paper that I published with a colleague a decade ago. And this is the data we have showing, just for Florida, this was the Gainesville area, that that Quercus virginiana was flowering earlier than Quercus geminata, and that probably allows them to stay separate species instead of, if they overlap too much in flowering times, they're going to share genes a lot more. And that might be an explanation for why they have distinct leaf morphology. And that's what you see here. It's a morphological index that combines a whole series of different traits we measured about the leaves. And when we combine them all, we see, yes, they actually have pretty different distributions of their traits. And Quercus geminata just looks different than Quercus virginiana when you take all those traits together. And this is genetic data taking a certain number of genes throughout the genome. This was not that fancy. This was a while ago, but what you see, the black, the y-axis shows the proportion of ancestry, and it says, okay, if it's all black, then it's probably one species. If it's all gray, then it's probably the other species. So all black would be Quercus geminata, all gray would be Quercus virginiana. But if it's half and half, then it's a hybrid, or there's been introgression, so they're sharing genes. And so all of these bars represent a different tree, and these are the southern ones in that says S here. So that's the southern part of the distribution in Florida. And then as you go towards the middle in both directions, it gets more northward. And here is a region in North Carolina where we think they're hybridizing, or there's been some introgression that we've definitely noticed. And you get even some misidentification of species because you can't quite tell from phenotype who's who. But so we're trying to test, but we don't have enough data at this point, the extent to which you get more introgression farther north because of season length, and whether there is actually evidence for more overlap in flowering time farther north. And it would be exciting to figure this out. So I'll pass to Ian now.
Well, I'm going to first ask Erin real quick, do I have time to have a couple slides, or would you like to continue on?
Yeah, go ahead.
Okay, great. And all right. I'll assume that everybody can see this unless I hear otherwise. But so Andrew and Janine presented a little bit on the importance of phenology for integration, which is basically to say if two plants don't flower at the same time, they can't mate. And that's really important for these species that do intergress and have overlapping ranges. There are a bunch of other things that we can learn from having detailed information about oak phenology. And part of that is just understanding the variation in events throughout the season for this really broadly ranged species. We can start to forecast when phenological events happen in the north or in the south across the range of bur oak and these other species that along with it. Something that's really interesting to me with this is that people plant these species all over the world. You can find, I've seen Quercus macrocarpa throughout the United States. I've seen it in Europe, and I've seen pictures of it in places like Australia. Do our estimates of phenology taken from places like Illinois or Indiana hold up when you go to Australia? And I think that's a question that we don't really know. Like, what is the scope of predictions of phenology? Another place where there's a lot of variation in phenology in oaks is at the very small scale. So this is a picture, it's actually of valley oak, a white oak in California, taken on a single day. And obviously these two trees that are right next to each other are at very different phenological stages. These are trees that probably do not mate with each other because by the time one is done flowering, the other one's not yet begun. So understanding that very low, small scale variation can be important. And specifically for some of the things that oaks do. So oaks are famous for masting. People have probably seen big bumper acorn crops in some years and really lousy acorn crops. There's almost none in other years. Something that people have found with masting is that those big bumper crop years are often years where you have a really fast spring. That's to say there's not very much variation in bud burst among trees. And the idea here is that if you have a fast spring, then everybody's kind of sharing pollen a bunch, and times are good for mating in oaks, and they produce a big acorn crop. And in those very drawn out years, maybe the opposite is true. So that's one hypothesis out there linking phenology to this really crazy big scale phenomenon of bumper acorn crops. The other really cool thing with oaks is that they're just foundational species. There are hundreds and thousands of animals that rely upon these trees as their home, and often it's their only home. This is a mural just to demonstrate of some of the various creatures living up in an oak tree. And some of my favorites of those are these small insects, gall wasps, which often are a spark of imagination for me, that they're so bizarre. They're these little insects that are able to figure out a way of tricking the plant into making them homes by somehow manipulating how a plant develops. But to do this, they have to get on that plant at a very specific stage. You can imagine that a gall wasp that's trying to coerce a leaf into some other way of development to make these bizarre structures where they live, have to get that leaf at just the right point in time. So understanding how phenology plays into the communities of these different animals that really rely on the timing of leaf set or other phenological events is something that I think is a big deal for understanding the big communities of animals up in oak trees. So with that, I will hand things back to Erin.
Thanks so much to all of you. That was a great overview of why the data that you all are going to collect as part of this campaign are so important. So thank you. Some of you might already be familiar with the National Phenology Network, but I just wanted to run through a little bit of an overview of what the NPN is and what Nature's Notebook is. So the USA National Phenology Network was established back in 2007. At the time, there really wasn't a way to have standardized phenology data collection to support research like what we've been hearing about. So we were set up back then, over 10 years ago, and the primary way that we were hoping to collect phenology data from across the country is through our Nature's Notebook program. And so as you can see on the map here, we have lots of different people that are participating all across the United States, collecting data in these different points here. And you can see by the size of the circles, kind of the places where we have a lot of data coming in from, mostly following the kind of urban centers and along the coast especially. So I would be curious to hear from you all if you are already collecting phenology information. I'm going to launch a poll here. And so if you're already a Nature's Notebook observer, let us know, or perhaps you're collecting phenology data for a different program. Maybe you keep a nature journal. Maybe you notice changes, but don't always write them down, or maybe you don't yet track phenology. So I think you can select any one of these. And I'll just give it a little bit of time so people can answer. We've got 70% of people have voted. Just give it a couple more seconds. All right. So it looks like we have quite a few people are already a Nature's Notebook observer. And then we have some people that do notice changes, but don't usually write them down. And then some people don't yet track phenology. So lots of potential to get you all involved in doing this program. So one of the things that we like to focus on in Nature's Notebook is suggesting certain species that are of particular interest to researchers or natural resource managers. And these are what we call our Nature's Notebook campaigns. So there's a link here, and I think Samantha's going to put it in the chat for us. There's a lot of different campaigns that you can join, so I invite you to see if there are other campaigns that might be in your area that you'd be interested in. All of these campaigns are inspired by a research question or a need for data. And so we have a lot of different types of organizations and partners that we work with to try to leverage all of the great observers that we have across the country who are able to collect data in their own backyards. We found that it's just such an amazing resource to be able to call upon all of you all who are keeping track of things happening where you live. It's just a great amount of valuable data that really all these researchers wouldn't be able to collect otherwise. We also make sure that all of the data that are collected by observers are made freely available. One of the ways that you can explore all of the data that are collected is through our visualization tool. So we can put the link for that in the chat as well. So all of the data that you submit automatically go into our database, and they're freely available for download. So we have a lot of different people that are accessing the information that you are recording and finding out different things with your data. Our visualization tool has a set of tutorials called the seasonal stories that you can look at, where you can dive right into looking at the data through the Data Explorer tool. And we have a lot of different people that are using the data. We're over 100 peer-reviewed publications at this point using Nature's Notebook data. Here's just a sample of some of the really different types of studies that have used Nature's Notebook data. And we do summaries about every other month of some of these articles. So I invite you to visit that NN vignettes page, which we'll also put in the chat. And that is where you can kind of learn about these different research articles that have come out. And we make short summaries that describe what the research is all about, what the findings were, and why the data are valuable. So you can check those out. All right, so let's get into the specifics of Quercus Quest. So I'm just going to run through briefly how you can get started with this campaign. So basically, the steps are you need to create a Nature's Notebook account. You'll add a site, which I'll describe what that is. You'll add at least one of our Quercus Quest oaks. You'll record data on your oaks, and then you can also sign up for our campaign emails. Even if you don't sign up for our emails, your data that you submit on these oaks will automatically be counted as part of the campaign. But it's important to sign up for the emails if you want to stay up to date on the results. We'll share back with you how the data are being used. So we'll describe how you can do that as well. All right, so if you are not yet a Nature's Notebook observer, basically you can go to the Nature's Notebook website and go to that Become an Observer link. When you set up an account, you can either do this on the website or you can use our app. You can actually do all of it on the app now if you like, if you don't want to have to use the web browser. So you can just go to any of the app stores and find the Nature's Notebook app there. Or you can go through the website. So I'm just going to run through what it's like when you sign up through the website. So basically, you just need a couple pieces of information. You put in your username, you create a password. You can skip the partner group section unless you are observing as part of an organization that's doing this together. If you're just doing this independently, just skip past that part, agree with the terms of use, make sure you're not a robot, and then go to create new account. And then you're going to be prompted to set up a site. And so what a site is typically the place where you're going to be making your observations. There's some considerations for a site. We want to make sure that it's convenient for you. So even if you could pick a site that's the best possible example of the most natural area, but it's really far away from you, you're probably not going to be very likely to go there. So we want you to pick something that's convenient. This can be your backyard. It can be some park that's near you, a trail that you like to go to. It could be near your office, just a place where you know you're going to be going pretty frequently. It's great if you can pick a site that's pretty representative of the surrounding area. So if your backyard is kind of urban, but you're in an urban area, then that's representative. If you want to go to a more natural area, like a park or another area that's a public land, you can pick an area that's just representative of its surroundings. We try to have the sites be pretty consistent in terms of habitat. So in this example graphic here, if you were in an area where there was deciduous forest and then evergreen forest, you might want to have different sites. So you can have more than one. If you had an area where there was a meadow, you might want to have that be a different site. So just try to make sure it's mostly uniform. And then as far as the size, it can really be whatever makes sense for you. If you're going to observe one oak, that can be your site. You don't have to worry about making it bigger than that. It's just that's the size of it. If you want to observe multiple oaks that are spread out, then it would encompass all of those oaks. Once you set up a site, you'll be prompted to add a plant. And so for this campaign, we have these different species are the oaks that are part of the campaign. We heard about these and why they were selected earlier from Andrew and Janine. So when you add a plant, you're actually going to be observing that same tree over time. So that's something that's really important to remember. You want to tag that tree in some way or remember where it is, and then go back and observe that same tree over the course of the year. And we do have this list of oaks also on the campaign landing page, so you can refer back to that as well. Once you add a plant, then you'll get to this third step in the sign up process. We'll let you know that we have some resources to learn how to observe, and then you can also print your paper data sheets from here, and then you can go to what is called your observation deck. So your observation deck is going to be the main place that you will do all the things you need to do on your computer to observe and enter observations. So you'll see up at the top here, we have some, this is our observer certification course, which I'm going to talk about in a bit. We definitely recommend going through that. There's also some other resources here. You can add things onto a calendar to actually visualize your own data. And then we have badges that you can earn as well. And I'll also talk about those later. But really the main part of the observation deck is down here at the bottom. So here you'll see your list of sites. If you're part of a Nature's Notebook group, you'll also see your groups listed. But if you're only observing independently, then you'll just see your personal sites. Then you'll see your list of plants that are at your site, a place where you can print out your data sheets. And then here's where you can enter those data sheets online. And you can also download your data here and then get into the visualization tool to look at your own data from here as well. All right, so if you are already a Nature's Notebook observer, then all you need to do to sign up for this campaign is to add one of the oak species from the list. So you can add that to one of your existing sites if that makes sense. If it's going to be your backyard and you have an oak, you can just add the oak by going to this link right here, add or edit plants. Or you can set up a new site if you feel like it's in a different area, it's a different habitat or something like that. You can add a new personal site and make the oak at that new site. All right, so let's look a little bit more closely at the data sheet that we have for the oaks. And I think most of the oaks have the same data sheet. So this should apply for all of the species. So each of the data sheets is basically a series of yes or no questions. So we have, do you see breaking leaf buds? And then there's a Y for yes, an N for no, and a question mark if you're not sure. For each of these questions, we have what is called a phenophase definition. And that is a very descriptive set of words here on the right that describe exactly what we're asking about for that. So you don't have to guess at what a breaking leaf bud is. You can just read that it is one or more breaking leaf buds are visible. A leaf bud is considered breaking once a green leaf tip is visible, but before the first leaf has unfolded to expose the leaf stalk or petiole for leaf base. So that really describes exactly what you're looking for. So definitely refer back to these. These definitions are on the data sheets, and then they are also on the app. For each of the questions, there's a little eye, and you can tap that on the app and then see those definitions. So the phenophases that we're especially interested in for this campaign are the beginning leaf stages. So we want to know when you see breaking leaf buds and leaves and increasing leaf size. We also want to know about the flowering stages. So when do you see the flowers or flower buds, open flowers, and pollen release? And you're welcome to report on all the other phenophases too. You can report on the colored leaves and when they fall. You can report on the fruits, the acorns as well. But the ones that we really want to make sure that people record if they can are these ones here for leafing and flowering. So I wanted to describe just a little bit more about how these phenophases can overlap, because I think this is something that can be kind of confusing sometimes. So when we're talking about the leaf phenophases, the first thing that you'll report at the beginning of the year is breaking leaf buds. That's when you first see that green leaf tip start to emerge, but before you see that leaf stalk. Once you see the leaf stalk, then you have a leaf. And so you start reporting leaves. And at the same time, while those leaves are still getting bigger, you'll want to report increasing leaf size. But you may be reporting all three of these at the same time. So if you have breaking leaf buds on the tree, you have some of the leaves have emerged, so you have the leaf stalks, but you still have some that are breaking, and you have some that are increasing in leaf size, you would be reporting a yes for all three of these at the same time. And then once all of the little leaf buds have emerged so that you can see the leaf base, you would stop saying a yes for breaking leaf buds. So you would be saying no for that, but you would continue saying yes for leaves and then also for increasing leaf size until all those leaves were at their full size. And so it can kind of sometimes take a little while to get to know your tree before you feel really comfortable with this. You might have to go through a full season before you know what the leaves look like at full size. And that's totally fine. That's why we have the question mark on here. So you can always report that you're not sure. And also you can skip any of the phenophases that you're not comfortable with. So feel free to just skip things you don't know about, and you can always report on them later once you know your tree better. So in a similar way for flowers or flower buds, these also have some overlap. So you would first start reporting flowers or flower buds when you see the first flower structures appear. And then once those are open, you would continue to say flowers or flower buds in addition to saying open flowers. And then pollen release would also, if you're able to see that, you would be saying yes for that, as well as all of the other flowering phenophases at the same time until all the flowers are dried up on the plant. So let's go through an example here for bur oak. So I wanted to look at these pictures because the oaks are a little bit complicated when it comes to the flowering structures. So oaks actually have what are called catkins for the male flowers. And these are flower structures that start out kind of bunched up, and then as they're opening, they expand and kind of hang down from the tree. So we would have, this is what you would first see at the beginning of the season. And you can see we also have a leaf structure that's appearing here. And from what I can see here, it looks like you can't quite see the leaf stalk. So I would call this breaking leaf buds here, as well as seeing some flowering. So these are the male flower structures. There's also female flowers that are separate, that are on the same trees, but these are often very hard to see. They're very small. And I think the researchers that are on here can correct me if I'm wrong, but I think they typically occur high up on the tree and they can be kind of hard to see from the ground. So you may never see the female flowers, and that's okay. But when you're considering the questions on the data sheet, any flowers that you see count as flowers. But more likely you're going to be seeing these male flowers are going to be more obvious. So if I was going to answer the questions on the data sheet for this one, this picture, I would say, yes, I see breaking leaf buds, but I don't see increasing leaf size or leaves because those are both leaf phenophases, and it's not a leaf yet because you can't see the leaf stalk. I do see flowers or flower buds. They're tightly bunched up, but I would not call these open flowers because they haven't yet expanded as they are in this photo here. For this one, you can see that actually this might be wrong. This might actually be a breaking leaf bud. So you might still say yes for breaking leaf buds. I didn't really notice this one here, but this one is actually a leaf at this point because you can see that leaf stalk, and then you can see some other leaves here. We're kind of limited in what we're looking at in this photo. So I would say a yes for increasing leaf size and a yes for leaves. I would say a yes for flowers or flower buds, and also a yes for open flowers because these catkins have expanded. For the last ones, we can't really see what's happening with the leaves because this is very close up. So I would say question mark unless I was actually there and I could see what was going on. For flowers or flower buds, I would say yes, but I don't think that these are open. I don't, maybe the Andrew, Ian, and Janine can correct me on this, but I don't see any reproductive parts visible here. So I would probably say a no for this. It could be that there are also some male flowers, and that would make it more obvious that yes, you see open flowers, but from what we can see in this photo, I think that these are closed, but they're very, very small for the female flowers. So when you are recording the data for Nature's Notebook, you can either use paper data sheets or the Nature's Notebook app. And you can see we have this list of questions here, just like we had in the previous examples. So you would have a bur oak, perhaps, that you have in your backyard. You would be going back to the same tree. And each time you made a visit, you would be answering the same series of questions. And we're very interested in when things are not happening, in addition to when they are happening. So make sure that you report those no observations if you're sure that that phenophase or life cycle is not happening on the tree. We also have a comments field here. And so that is where I would recommend if you do see things like Ian was talking about with the galls or any sort of species interactions happening, you can always put that in the comments field for your observation. So with the paper data sheets, you would be taking those observations on paper and then entering them on the website, or on the app, you would just be answering those questions and then hitting save data, and the data are automatically submitted into the database. I wanted to just mention a little bit about frequency. So I know people wonder, how often should I go out and observe? We would love it if you could observe a couple times a week, especially in the period where things are really happening with your oak. When it's more of the dormant season, when there's not a lot going on, it's fine to only observe every few weeks or every month. But once you start seeing activity happening and those little buds starting to be active, that's when hopefully you would do more frequent observations. And why that's important is when you're recording those no observations followed by a yes, that really gives us a better precise estimate of when that thing started happening. So if you were only reporting every 6 days, there's a bigger window of uncertainty here when we don't know when that thing started happening. But if it was every 3 days, we know it was only 3 days of uncertainty in between that last no and when you actually started saying yes for that different life cycle event. And wanted to also mention that we have, in addition to the yes or no questions, we have intensity questions, which are questions about the degree to which that life cycle stage was occurring. And so for something like flowers, we're asking you to estimate, putting your estimate into these bins here about how many flowers or flower buds do you see on the tree, and then what % of those flowers are open, and then how much pollen is released. And so these are always optional. If you feel like you're not comfortable, then you can just skip those. If you do answer like a yes for flowers or flower buds, you'll be prompted to say how many were present, but you can always just say not sure or ignore that. But if you feel like you can't estimate these, it is just an estimate. So you can just try to put your estimate into one of these bins. And that can really help to get a sense of how much of that phenophase was occurring. And so, for example, with the open flowers, this really lets us see not only the no's and yeses, but when the peak happened. So when were the most flowers open on the tree? And that's really important information for knowing, when the potential for pollination was the highest, when the flowers were open. And for something like the leaf canopy, when was the tree most full of leaves? All that information can be really important as well. And if you track your oak for at least 6 different weeks within the year, you can earn your Quercus Quest badge. And so these are a series of badges that you can see on your observation deck. So I encourage you to watch out for that and try to get your 6 different observations in 6 different weeks of the year. All right, so I have a couple pictures I thought we could kind of try out making observations. So I'm gonna have a poll here, let's see. And really, when you're considering the questions on the data sheet, really consider each question separately. Don't try to figure out, okay, what are all the things going on the plant right now? Just try to go one by one and answer the questions individually. So I'm gonna have just a single question I'm gonna ask you here. For this picture, do you see breaking leaf buds? Yes, no, or not sure. And you can use that phenophase definition to remember what exactly we're talking about by breaking leaf buds, and try to do your best to answer what you see here for this white oak. And we've got about half of people have voted. I'll give it a little bit longer. All right. So most people said yes. We had a couple people say no. So let's look at the definition. So what we're talking about is when a green leaf tip is visible, but before the first leaf from the bud has unfolded to expose the leaf stalk or leaf base. And so this is a little tricky because it says green leaf tip, but this plant looks red. So that is something that can be confusing sometimes. We are talking about the fresh new leaf tip, and sometimes it's not always green. This is a definition that we use for all the different deciduous trees, and a lot of them are green, but there are a couple that have a reddish looking leaf bud. So even though this is not green, it is a leaf tip that's emerging, and we can't see the leaf stalk yet. So I would say this is a yes for breaking leaf buds. So I know we have another phenophase for colored leaves, and sometimes people think this looks like a colored leaf, but really the colored leaves phenophase is for that late season color when you have full leaves that are the full size that are turning color at the end of the season. So don't ever report colored leaves for this kind of leaf. This is a breaking leaf bud right here. All right, so one more question for this picture. Do you see leaves? All right, we'll give it a little bit more. Most people have voted. All right, so we have most people said no, and more people than last time said yes. So we don't yet have a leaf here. So again, this is not, you can't see the leaf stalk or petiole or leaf base here. So this is not yet a leaf. This is a breaking leaf bud. So if I was going to answer the questions on the data sheet, the only one I would say yes to is the breaking leaf bud. Once we see the leaf stalk emerge, then that is considered a leaf. And then we can report leaves and increasing leaf size until all the leaves are the full size. All right, so we have one more question here, or 2 more questions about this next photo. So this one is about whether you see flowers or flower buds. And again, this is for bur oak. Hopefully this isn't blocking your screen. You can move your poll around if you need to. But again, look back at the definitions. For our flowering phenophases, we actually have what we call species specific definition. So this actually says for Quercus macrocarpa, which is bur oak. So we're asking you to look exactly what we're talking about with this specific species. We also have that information for the fruit phenophases as well. All right, I think most people have voted. I'll give it another couple seconds. All right, so we have almost everyone said yes to flowers or flower buds, and a couple of people said no or not sure. So yes, so for the flowering phenophases, flowers or flower buds is any sort of flowering structure that's on the tree. So even if you didn't know whether or not these are open, you know that there's some sort of flowers or flower buds, so that is a yes. And then one more question about this picture. Do you see open flowers? All right, I think about everyone has voted. All right, so most people said no, a couple of people said yes. So I would also defer to our researcher friends here. I would say this is not yet open flowers because these look still very bunched up to me. They haven't extended into those long catkins. So I would say this is a flower bud, but it's not yet an open flower. So I would say no for open flowers until you really see them extend down like an accordion and open up, and that is what is considered open flowers. And this kind of describes that in the definition. The male flowers will open once the initially compact catkin has unfolded and it's hanging loosely. So that's the part you want to look at here. All right. Hopefully I shared that already. Okay, so as I mentioned, if you are contributing observations for any of the oaks that are on this list, your data will be counted as part of the campaign. But if you would like to receive messages, which will be filled with lots of tips, we'll have some prompts for things to watch out for on the different species. We'll have some resources for you. And then of course, we'll have lots of results of what you are reporting and how it compares to what others are reporting across the Eastern United States. Then please sign up for messages. We'll be sending those out, the first one in the next couple weeks, and then every 4 to 6 weeks throughout the season here. So I think Samantha will put that link in the chat as well. It's also on the Quercus Quest landing page. Okay, so just a couple more things to go through, just some special considerations. So I know that sometimes it's hard to identify oaks. So I think you can use mostly the leaf shape and the acorns for identification. There's also this great field guide. And so Samantha's going to put that link in the chat as well. And that can kind of help you to look at different features to make sure you can identify which species you have. Also note that even if you have a tree that's mature, it might not always have acorns. So if you are tracking that fruit phenophase, don't be too concerned if you don't see it happen on your tree every year. It may not. Also note that it's the female flowers that are going to have the acorns. So if you've been watching those male catkins and then nothing ever happens with them, the acorns might be in a different spot on the tree. So just don't expect all the catkins to turn into acorns. That'll be the little female flowers that are going to be producing the acorns. We're also very interested in if you have more than one of the species on our list. Those sites are particularly valuable. So if you can observe more than one oak species, that would be very great. But of course, just one species, if you only have one, that's totally helpful as well. We are also interested in the site conditions that you have. And there are certain questions that you can answer about your site. So I'm just going to show you where you can do that. And so this is either if you already have an oak that you are observing, you can add this in later, or if you're adding a new one. Go to your edit site link on the bottom of your observation deck. You actually can't do this on the app, you have to do it on the computer. And then fill in the details here under additional information. You can tell us more about what your site looks like, and you can go to this plants tab and answer even more questions. All that extra detail is really valuable for us to have. And then also when you add your oak tree, you go to the add or edit plants, and even if you have one already, you can go and edit it. And then please look at these details here about these questions, the shade status, whether it's wild or watered or fertilized. If you know the planting date, that's helpful as well. So if you can fill in all those details, that would be really helpful. So I mentioned we have these phenophase definitions. Make sure you keep referring back to those. We also have a botany primer that I welcome you to check out, and Samantha will put the link for that in the chat as well. It has a lot of great pictures that kind of explain catkins and different aspects of botany. We also have an observer certification course, so I welcome you to check that out. You can find the link to it on your observation deck here. And then it has many different modules. The basic module, the first one is just kind of how do you do Nature's Notebook, so definitely take that one. And then there's some more advanced ones as well that go through information about the phenophases, and we're working on one right now for helping to estimate intensity. So that's definitely a good course to take. I also wanted to mention very briefly, we have what are called local phenology programs. And so I'm not going to get too much into those right now, but if you are observing as part of an organization, we can set up a local phenology program for you where you can have multiple observers tracking the same plants at the same locations. So please reach out if you'd like to learn more about that. We actually have a certification course for local phenology leaders that is actually going to start in a couple weeks. So feel free to reach out if you have questions about that. And Samantha will also put a link in the chat about the application for that. All right, so just a little recap. I want to make sure we have time for questions too. So just to run through again your steps for getting started. Create a Nature's Notebook account, add a site and at least one oak, add your details about your site and your oak, sign up for the messages, make sure you take advantage of the training, and then record your observations at least once a week if you can, but really we're interested in any observations that you can make. And then feel free to reach out with any questions you have. And we also have some time now for questions for either Samantha or I or any of our researchers.
Hi, yeah, we had one question earlier about if we're ever going to do any observations outside of the United States, or are we just in the United States right now?
Yeah, so technically you can observe anywhere. If you have the species that are on our Nature's Notebook list, there's nothing that stops you. We do have sites in Canada and Mexico, especially, and also in some other countries where they have like a botanical garden and they have some of our species. But I don't think our app works in Canada, so that is one limitation there.
And then we had one observer note that she saw that Quercus bicolor was not included on the Nature's Notebook yet, but it's included in the species list. Is that one that's going to be added later?
Yes, thank you. I forgot to mention that. So we're a little slow on our species additions this year. Typically we have them up in February, but we do have, I think it's something like 6 of our 9 species are getting added right now. I think they'll be up online by Friday, but it might be Monday that they're up. But we will be sending out an email about that once they're up. Thank you. Yeah.
And then we had one more question of what happens if they identify an oak as a white, but later determine that it's a burr, or so basically you misidentified it but figured it out later. How's that something that they can fix?
Yeah, so that's not something that you can fix on your end, but if you reach out to us, we can change the species for you. And it should be fairly straightforward because these oaks have the same protocol. So just reach out to us. You can email me or whatever email you find on the contact on the website, and we'll get that changed for you.
And then I had one more question about polls. Some people were wondering about the results of the polls. Do they show up on the webinar, or is that something that we send out?
That's a great question. I don't know. I'll have to see when I put the recording up if they're there, but I can put them in the follow-up email when I send that out to everyone who registered. Yeah, any questions for our researchers? Because we have them here and available if you're curious about anything. I know that they covered a lot of different things.
I should mention we have a few of our other researchers here whom I didn't notice. We have Lucy Schroeder, who's from University of Minnesota. We have Ellen Whittemore from the U.S. National Arboretum. We have Heather McCarthy from Oklahoma as well. And who else are there any of our others? Oh, we had Mira Garner from the Morton Arboretum. Did I miss anybody else from the group? Okay.
Yeah, anything else that any of you want to add, feel free to jump in. It's so nice to have such a big research team that we're working with.
We did have one question that popped up. So when different species of oaks hybridize, are their offspring going to be viable?
Sure. Yep, I could take that. Their offspring are viable. They show decreased fitness. So if you look at the acorns, they may not be as fit, but they do produce offspring, and that back crossing. So you get that first generation hybrid, the back crossing to the parents. After a few generations, you may not be able to tell that there was any hybridization at all. But alleles, gene copies from the one parent that it doesn't look like, that these offspring don't look like after several generations, may be dragged in. And so it can carry these are potentially beneficial to the offspring. And that's part of the whole question we're interested in, is how can that dragging of alleles from one species to another influence all the communities of organisms that depend on oaks?
I'll also mention, because this is a new campaign, we are still looking for a lot of photo resources. So if you are able to take some good quality photos while you're observing your oaks this year, please send them to us, because we would love to make some photo resources to help other observers. And we're especially looking for the kind of up close, high resolution pictures of the breaking leaf buds and the flowers, and when the flowers are open for the different species on the list. So if anyone likes to take pictures, that would be very helpful.
And we just had another question come in, Erin. When does the monitoring start and end? Is there a certain time that they should be monitoring these oaks, or can they do it year round?
Yeah, so you can definitely do it year round. I think Andrew or Ian, I don't know if you want to answer about the general timing of when things happen. I think it's like in the next few weeks for most of the oaks, but some of the southern ones are probably already active.
Yeah, in the Chicago area, we will get flowering beginning probably, I think, at the end of the month. Boy, I'd have to look back at my notes to see. Anybody else can speak up if they have. Oh, thank you, Christy. Current forecast is early April.
All right, and then Erin, for submitting photos, do you want to do them attached to email or use the smug mug?
Yeah, if you know the process for doing the smug mug thing, then definitely you could do that. But if you're not sure, you can just send them to me or send them through the contact form on the Nature's Notebook website, and we'll get them to the right place. And I think I saw someone asking about that mural photo, Ian. So I don't know if you could share where that is located.
Oh yeah, it's actually a mural on a public restroom in Davis, California. So yeah, it's a really cool mural. Yeah, I'd be happy to, maybe I could send that to Erin, who could distribute. I don't know if I have a high quality picture, but I have a picture at least.
Cool. Any last questions?
Hello, I am Boyd Strain.
Uh-huh.
I joined you about 15 minutes before the end of your period. Is this available on recorded so that I can see the entire thing?
Yes, yeah. So we're going to post it on our YouTube channel, and then I'll send out the link in the follow-up email, and that's going to go out to everyone who registered, even if people weren't here. And then we can also put it in the campaign email that we send out in the next couple weeks. So yeah, there'll be multiple places you'll be able to see it.
Okay, well, I really want to participate. I'll try to catch up.
Great, thank you. All right, University of, the YouTube, it's our NPN YouTube channel. which is linked on the website, but I'll send out the email. That'll probably be the most obvious place to find it. And then we'll have it in the campaign emails. Oh, and someone just asked about the identification guide. Maybe we can post that link again.
I'm going to post it again right now.
Awesome. Well, thank you so much, everyone, for being here. I don't know if our researcher team has any last thoughts, but I really appreciate all of the great questions and engagement, and we are very excited to get the campaign up and running in the next couple weeks. So please sign up for the campaign emails, and we will start sending you those updates.
Thank you, everybody, for all your help. And if you have questions about any of the things we talked about, please feel free to reach out to any of us. We're all easy to find. I'll put my email in the chat right now.
Thanks.
Thank you all. And thank you, Erin and everybody at NPN.
Thank you.